the project is called Food for Me, and it's personalized nutrition and integrated analysis of opportunities and challenges in personalized nutrition. And I got these slides from Bruce German, and this is what you get when you drive on the motorway. And every single driver on the motorway gets the same information. They're told about low bridges, high bridges, and what have you. And each one has to deal with it. But there's a little bridge in a village in California which says, if you hit this sign, you will hit that bridge. So the driver now has personal information. If he hits this sign, he's going to hit the bridge. So this is the idea that you will empower people to make decisions based on a knowledge of their bodies in the same way that this guy can make a decision based on his truck. Now, an awful lot of people are under the impression that everything that has to do with personalized nutrition is genetics. And a very powerful message that comes from Food for Me is that that's not the case. We have personalized genetic information, but we also have personalized dietary data. In other words, we can help a person to understand their food intake, their nutrient intake, the bad choices they made and the good choices they made. And then we have what's called personalized phenotype data. And phenotype data is about when you look at me and you see a big guy, a fat guy, a slightly graying guy, all that. And then inside I have all these molecules, and that's my phenotype. What you see and what the biochemist can determine inside me. So all of these three are important. And so the way we look at it is there are three levels of personalized nutrition. The first one is dietary analysis. The second type is phenotype. And the third one is genomic. And as I'll point out to you, the third one, which is what most people think of, is the least developed. It's by far the least developed. So John will take you through a proof of principle study that we're dealing with, in which people go on the internet, they enter the data into their computer system, and they receive feedback. And the feedback we give them is visual. We don't give them quantitative information. We use a traffic light approach. So, for example, you can be green in relation to vitamin C. And there's quite a wide range there. You can be in the red side because you've too much, or the red side because you've too little, or you can be in the orange side. And that's what our volunteers, we have 1,200 volunteers taking part in their study, that's what they get back, and they're told where they lie. We want ultimately to be able to talk about automating this. So an individual goes on the internet in personalized dietary analysis, puts the information up, we give them, or we analyze their nutrient intake, and we compare it to a standard. And then based on their food choices, we come back with messages. So if, for example, they have a high level of saturated fatty acids, we can examine the sources of saturated fatty acids in the person's diet, and we can give them, if, for example, it's coming from too much dairy products, they'll get a message about food group 1. If it's coming from some other source, they'll get a message for food group 2. But ultimately, the idea is to automate the whole thing. Now, one of the problems we have is that if you want to look at a person's diet accurately, and when I say accurately, I mean to the first decimal point, then you spend a lot of time gathering accurate information. We are using the traffic light system. So we're trying to get a balance between the ease of use and the accuracy of the data. And so for epidemiologists who want to work with this, they can't do what we're doing. What we're doing is simply putting people into boxes. Safe box, not so safe, and unsafe box. And so the ease of use is really, really important, because if a person finds it difficult to enter the data, they won't use it. My best example would be the NIH the Cancer Institute in the United States. And it took me, well, I'm not very good at these things, but it did take me 20 minutes to get past breakfast. And I gave up at that stage. So I would hope that in our system, 10 minutes would see the whole thing done. So the ease of use relates to, in our case, just traffic lights. But if you wanted it to be very accurate, you'd be into grams of this, that, and the other per day. One of the things we're going to do in this project is to look at meal coding systems. And here you see 8 photographs of meat, different forms of lamb and different forms of beef. Now, nutritionists would code all of these separately. We're putting forward the idea that actually, if you're talking about green, red, and yellow, and broad bands, you don't need to code them separately. The same would be true of vegetables. We don't need to code peas and beans and broccoli differently. And that's an area we're looking at. So the idea would be, in the long term, that an individual would go online and have a series of screenshots of different types of breakfasts. And they would pick the breakfast that suits them best. So they would click and drag it down. And then they would be given options of what exactly they meant with. And they would click, everything would be click and drag. And that's what we're heading towards. When we move to personalized phenotype analysis, there are a number of areas to look at. One is inside yourself. So this is Homer, and he's interested in his folate or his insulin, his glucose, and what have you. If you're going to do personalized nutrition, and you have a system which allows people to connect with you on the internet, they don't need to come to a special clinic, they don't need to travel anywhere, they just get on the internet and do it. What happens when you want to take a blood sample? Do they have to come to a clinic? Because if we want to know what's inside them, we need to take a blood sample. And the answer is, just as we can connect with people using the internet, the system we're using is not to take blood samples in this form, but it's to adopt the heel prick test that is used for babies. And so we have these cards that people use. And they get a video, and the video explains how best to give the blood sample. So, for example, it advises you to keep your hands down, keep them warm, and so forth. And I think it's true to say it has worked out wonderfully well, certainly everywhere. But we get 5 blood samples. We have, in fact, 2 types. We have a paper type for water-soluble vitamins, and we have a plastic type, which is under experimentation with our colleagues in DSM for fat-soluble vitamins. So we send these out in the post, and people do it and send it back, and so there's no need to go to a doctor. The correlations between the serum cholesterols and the dried blood spots are remarkably good. And again, for a specific vitamin like folic acid, remarkably good as well. This is just to show you one of our partners, Professor Hannelore Daniel from Technical University of Munich. These won't mean much to all of you, but I mean, basically, in one dried blood spot, this lady can do all this analysis. So the potential is quite remarkable. There are devices out there which people with diabetes use, and this is an example from Hannelore's work comparing in the red case, the red dots there are from the dried blood spot, and the closed circles there are from the diabetes development. And here you can see the time going across here up to 2 hours, and things which change dramatically are in red, and those which don't change dramatically are in green. And again, excellent correlations between actual whole blood and blood spots. Now, the problem with this is that when you go into a person's phenotype, is that if you have 3 categories called high, normal, or low, which is red, orange, and green, so to speak. And this is just for 5 metabolites, lipids, vitamin D, insulin, fatty acid. And then someone else can come along and be slightly different. And actually, the number of options for that, where we have 3 levels and 5 metabolites, is 243 possibilities. Now, it is absolutely and completely impossible for us to provide a solution to that. We cannot have 243 personalized diets. So one area which we're looking at within Food for Me is the possibility that although we talk about, at present, we talk about population dietary advice, and in personalized nutrition, we're always talking about individual dietary advice, is that we actually talk about metabotypes. Now, what's a metabotype? A metabotype is a pattern of metabolism which many, many people share. So this is an old slide. I've had it for, I think, about 10 years. I've never changed it. It's Croke Park on All Ireland final day. And what I'm saying is that you can take the Irish population and, basically speaking, with 90% confidence, you can reduce them to about 8 or 10 metabolic patterns. And if I can do that, then I provide a solution to 8 or 10 metabolic patterns rather than to 80,000 people. And this, Ben Van Omen, my colleague, has referred to this as the head and shoulders approach. That there's a variety of types of head and shoulders, not one for everybody in the audience, but there are groups, and you pick the head and shoulders that suits you. I want to give you an example of how this works. And we use a thing called cluster analysis in our data. And cluster analysis tries to find out who's like who. So the computer, I use the computer like as though it's a person. But their software picks an individual at random. And then it picks another individual and a third individual. And it says which of those 2 individuals are closer to this person. So it moves them. They bring in a 4th and a 5th person, and they say which is closer. And slowly they build up clusters of individuals, 5, 6 types of clusters. Now, we've looked at this cluster analysis in terms of its potential to contribute to personalized nutrition in this cluster approach. I want to give you an example of it from a study we did with vitamin D. We had 160 subjects. 80 of them got vitamin D and 80 of them got a placebo. And at the end of it all, we found no difference between the placebo and the vitamin D in terms of any of the measures we had of the metabolic syndrome. The reason why we did the study is there's a lot of interest in vitamin D and the metabolic syndrome. So we found nothing. Of course, the vitamin D improved in the people who got vitamin D, and it stayed constant in those that didn't. We then divided them into what are called deficient, insufficient, and normal levels of vitamin D at baseline. And of course, those that were deficient show the biggest response, and those that were normal show the least response, but still nobody improved their metabolic syndrome. And then we did some cluster analysis. We were able to identify 5 clusters based on the measures we had of the metabolic syndrome. And what we observed was these 5 clusters. So for example, cluster 1 is a reasonably large cluster showing high levels of low-grade inflammation. Cluster 2, definitely this is a group with the metabolic syndrome. Cluster 3 is a really healthy group. 4 and 5 we didn't understand. Just didn't get it. We didn't understand what they were about. But what we did find was that cluster 5, all the people in cluster 5 responded to vitamin D intervention. And so if we looked at vitamin D in that cluster alone, the more we changed the vitamin D levels, the more we lowered their fasting blood glucose levels. So what we did was, effectively speaking, a proof of principle where we showed that you could use cluster analysis to identify groups of people that behave similarly. And that is a major part of our research now, ongoing. And it's a big part of Food for Me. And there's a parallel project called NutriTech, which Ben Van Omen runs. And we have several people working in that area. So personalized nutrition, from a dietary point of view, no problems. We can solve that. Personalized nutrition from a phenotype point of view, we really have to put people into a certain type of category and then provide the solution. In addition to looking at blood samples, there are other things that you can look at with regard to phenotype. Your waist, your fitness, your weight, your height, your sleep patterns, your lung function, and so forth. And there are two very common phenotypic devices that we use: weighing scales and a belt. And the belt is by far the best. But Hannelore Daniel is spending a lot of time looking at all of these different devices. And one of the biggest partners, industrial partners, is Philips. And Philips have a major interest in this area. And this whole idea that you can empower people to measure their biology is growing at an enormously fast rate. So this is an area that we think is important. And so our volunteers, at least those in the UK and Ireland, are receiving from Philips a very sophisticated device for measuring their physical activity. And it directly connects with Eindhoven. And you measure the device, and then you connect it to the internet, and it goes all the way over to Eindhoven on the web. And a couple of sports scientists there come back with options for your fitness, how to get fitter, or how to exercise better. You pick the one you like, and you get this device you wear. And I don't think I have a photograph of it, but it's red lights across a little thing the size of a matchbox, half a matchbox. And as you accumulate exercise during the day, it goes from red to green, 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 green. And at the end of the day, it should all be green. But you can measure sleep patterns, you can measure blood pressure. Hannelore Daniel has these devices which she puts on subjects which measure glucose constantly. We actually have skin patches which measure vitamin D. So this is a huge industry. So these are just examples of the gadgets. This is the Philips that they wear, little device like that. This is for measuring sleep. This will be a thing in a pharmacy or in a doctor's suite, which measures bone density. And I like this one. This is a shirt worn. This is part of a Framework 6 program. This man is wearing a shirt which has nano devices, which is measuring his pulmonary and cardiac function. And it's transmitting it wirelessly as he sleeps in the hospital to the base station. So there's a huge future in this aspect of it. Now we move to the third aspect of personalized nutrition, which is genes. And I did say at the outset, and I'm slightly biased here, that I do think this is an area which needs huge amount of development. Again, can we connect with people without having them to come into a clinic? And the answer is yes. We stick this in the post, and they do their mouth swabs and send it back to us. So it, again, works on a mass basis. And we have variations in the sequence, so small variations make big differences, and that's why we're all slightly different. This is the percentage energy from dietary fatty acids, from polyunsaturates. And in this case here, where polyunsaturated fatty acid is low, there's no difference between these genotypic people. But where fatty acids are high, there's an enormous difference. If you happen to be this type of genetic person, and you happen to be put on a diet high in polyunsaturates, you will show a very high level of this undesirable profile. So you could say that therefore is very powerful information for an individual. Now the problem with all of that is the literature, the scientific literature, is awash with this information. But it doesn't prove cause and effect. This could be completely and utterly accidental. So I want to just show you a study from some of my colleagues in Trinity College Dublin and at the University of Ulster. And it regards riboflavin, a B vitamin. And it looks at a very complex phenotype, blood pressure. Blood pressure is not a simple thing. It's very, very complicated. And it looked at people who had genetic variations in an enzyme involved in folic acid riboflavin metabolism. And these were people who were already on drugs, so that they were not just diet. So the three things we're looking at here is high blood pressure, a genetic variation, and the intake of a micronutrient. Now, in 2004, they had an intervention with 1.5 milligrams of riboflavin for 16 weeks. They had an intervention group and a control group. As I said, 88% were on blood pressure drugs. 51% were achieving their blood pressure goal. And this is the genetic variation, and this is an enzyme involved in folic acid and riboflavin, and this is the variation here. So they had trouble publishing this, so they had to go a second time. So in 2008, they went back to the same groups and they swapped them over. And those that were the controls became the intervention, and vice versa. By now, 95% were on blood pressure drugs, 65% were achieving their goal, and they had exactly the same mixture. And what they found was that in the 2004 intervention, with the riboflavin, their blood pressure dropped right down. And then it rose back up again. And this group here, the placebo group, showed a very small but non-significant effect. But in 2008, when they were shipped over to the intervention, they showed an effect. Now, this is the only paper I know of in which a genetic relationship between diet and health has been proven. Every other one is associations. And that's the problem. So where's the business? And Yo is going to help us to find that one out. About 10 years ago, the people in the Institute of the Future at Palo Alto got it wrong. They suggested that by 2010, a third of us will be making choices on personalized nutrition, and they got it wrong. You can't have a Coca-Cola for every genetic variety. You can't change the eggs. So you have to change the choice of foods. And you could do customized supplements, but basically speaking, you really have to, you can innovate in new products, but you ultimately, in personalized nutrition, this is the way to go. You have to help the consumer to make the right choice. And you give them information about their diet, you give them information about their phenotype, and maybe one day information about their genotype. And with all of that information, you put it together and you put it in here. My vision of the future is that a person will go shopping and their food choices in the shop and even their food choices at work will be connected to a device at home that keeps a record of everything they've eaten. And in Philips in Eindhoven, they have a kitchen with a plasma screen like this. And everything that goes into that kitchen is known. And when somebody makes a breakfast cereal in the morning and they simply go online and say, I had a breakfast cereal, or whatever it is, it knows what went out of the system, so it knows what's left. It can offer you recipes, it can offer you menus, it can offer you all sorts of solutions. So solutions are what people are looking for. And that's really what personalized nutrition is all about. So, just going back to my last slide, level 1 personalized dietary analysis, immediately possible. personalized phenotype, probably we need to focus on clusters. I'm afraid that despite the popularity of the topic, the genes have a long way to go yet. So that really, I think, is all I have to say by way of introduction, other than these are the people that fund our work in personalized nutrition.